Hello, hello. Today we're going to be talking about what is a biology. Today specifically, viruses. This will be wrapping up our little module section thingy on nucleic acids. So, what is a virus? It's something that's inanimate. It's not alive. It can't even reproduce on its own. It doesn't even have sex. But it still sends us into a global pandemic and ruins all of our lives. So, viruses have this inability to reproduce by themselves, so what they have to do is infect a host to do it for them. So, some little nomenclature first of all. You've got a virion. That's just an infectious particle. That's just a single one of these viruses, essentially. And you have a host range. A host range means the types of hosts that these guys can infect and then reproduce these viruses. Then you get something called a phage. A phage is something that is just a specific type of virus that infects bacteria. So it's also usually known as a bacteriophage, but nowadays we just call it a phage. And a clone is a progeny virus. So you take one virus and then you infect a cell, then the cell reproduces a bunch of the same virus, and they're just clones of the original virus. I mean, like Boba Fett with all of the, clones in Star Wars. That prequel, that shouldn't have probably happened. But, anyways, got a virus here. It's a nice, beautiful, realistic drawing, not a cartoon. So, we have a couple of different things here. So, with the coronavirus, you might have heard something called spike protein, and that's been all the news back in the time, and that's this thing called a glycoprotein. On the outside of the virus, you have a glycoprotein that is shown and presented that is usually typical of just a specific type of virus. There can be usually like 1 to 2 of these glycoproteins that are usually presented and encoded by the virus itself. So how is this guy embedded on the surface? What even is the surface of the virus? That's the envelope. The envelope is just a lipid membrane, like the plasma membrane of the cell. And this membrane is actually unique in a lot of different ways, such as the incorporation of a lot of single chain lipids. So this lipid environment is what holds everything in. So then you have inside, you've got a capsid, this little octagon I got here, but they got so many different geometries that it's crazy. So this is a capsid, and it's essentially just a protein barrier that helps encase a nucleic acid, RNA. And why did I just say RNA or DNA? Because inside one of these viruses can be any type of nucleic acid, a DNA or an RNA. And one thing to note is that semantically, when a nucleic acid is inside the capsid, it's called a nucleocapsid. Fancy terms. So, got some fancy little other terms here. A prophage, remember what a phage is? It infects the bacteria. Prophage actually injects DNA and it's replicated in the host genome. And a template phage that associates with the cell, but it does not kill the cell itself. And remember, these phages only infect bacteria. So there are a couple different types of viruses, and typical virus just injects RNA, and it's all good. Well, all good for the virus. Injects it, and we're going to go through the little mechanism of how it does that and reproduces. That's the coronavirus. That's our least favorite little thing right here in the world. But you also have things called retroviruses and proviruses. A retrovirus has 2 identical RNA strands that are translated backwards into DNA, or cDNA, complementary DNA. We'll be getting into that in probably like the next video. So what the nucleic acid actually does, it encodes a reverse transcriptase. The reverse transcriptase, instead of going with transcription, going from DNA to RNA, goes from RNA to DNA, and then what happens is that DNA incorporates into the cell, and it integrates into the chromosome, and that usually causes some very, 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 very bad things to happen, like cancer, HPV. This retrovirus is HPV. Now, one thing to just remember about HPV, just a little quick note, is that HPV is a large-scale family of viruses. Almost all human beings have a strain of HPV. If anyone has a wart of any kind or like a skin blemish, that's HPV. But it's not cancerous, it's not bad, it's just a specific strain of the virus. However, there are several horrible, cancerous, terrible types of HPV that can infect the cell and cause cancer and horrible outcomes are caused from this. And then you also have a provirus, and a provirus is integrated, it's just DNA. So instead of RNA, you have DNA. So, now we know semantics, we know what a virus looks like, we know a couple of different types of viruses. But so, you're telling me it infects a cell and the cell makes it. But how does it do that? And that's what we're going to go into next. How this inanimate thing is able to ruin our lives. So, you got a virus that somehow gets into our body, somehow mysteriously by, I don't know, licking the boot of your shoe. So it comes in the esophagus and it finds some random cell and it then does a couple of different things. And it undergoes the lytic cycle. Now the lytic cycle is this guy, which is the process of which a virus replicates. Goes by these things: absorption, absorption, penetration, replication, assembly, release. And this cycle continues until either the virus is killed or the host is killed. So, first of all, adsorption. What even is adsorption? It sounds like absorption, but it's got a D instead of a B. And really it's just a fancy term for a specific type of absorption. So adsorption means it just kind of goes onto the surface and gets integrated with it. And then it gets absorbed using this thing called endocytosis, where a cell will actually take something from the outside and bring it in, and it will essentially encase it in a little vesicle, just kind of a circle of the cell membrane itself. And then what happens is, so you got penetration here. You've got it adsorbing onto the surface, going onto the surface, and penetrating in, comes into the surface, and then we have it fuse with that vesicle itself and release the nucleic acid, this RNA or DNA, into the cell. And so that RNA DNA is out in the cell. It's this wiggly boy that's just going and doing its thing. So in a lot of different in immunology immune responses, you can have something like C gas, a specific type of protein, bind to this viral DNA or RNA and then cause an interferon and cause the cell to die, so this way you don't die from the virus itself. But if that doesn't happen, you have this nucleic acid go and become protein. So it'll go and find ribosomes in the cell, on the cytosol, and make a bunch of proteins, and also go to the ER and make a bunch of glycoproteins. That's just one thing to note, is that membrane-bound proteins are made in the endoplasmic reticulum and centered out into the plasma membrane through some weird, fancy little pathways that we'll talk about in a while. But that's just one thing to note. The endoplasmic reticulum, it's also located right next to the nucleus of the cell. So this guy gets made into a bunch of proteins, and also this guy also gets, replicated. He makes a bunch more of itself by using its own proteins to reproduce this mRNA sequence or nucleic acid sequence. And so then you have all these guys come together and assemble, and so then they go out and become a virus again. And what happens is the process continues, goes to a different cell, and continues until we have a bunch of viruses killing the entire cell. Now, why is this called the lytic cycle? What even is a lytic? What is a lysis? And lysis is essentially when you break open the cell. So what happened, what the theory originally was that you have a bunch of these viruses just accumulating and making themselves until the cell broke open with all of these viruses on the inside, because it just can't take the strain of holding 100 viruses within it. But we found out that that's typically not true, and but they stuck with the name. Usually just you have a bunch of different viruses kind of continually get released from the cell until the cell is like, I can't do anything else, and just kind of dies and breaks open, doing lysis, but not in this huge, amazing event. So that is viruses in a nutshell. You got a virus that's surrounded in an envelope with glycoproteins around it that are the proteins that are presented. And so within it, it has a nucleic acid that is surrounded by a capsid, which is a protein coat that helps surround it. You have a lot of different types of viruses. Specifically, you've got a retrovirus that has RNA goes into DNA and usually causes cancer of some kind. You have proviruses that have DNA within them, and just regular viruses that have RNA within them. And they undergo this lytic cycle where the RNA goes into the cell and it makes a bunch more of its own RNA and a bunch of proteins that get associated with the virus. The virus reproduces and reproduces until the organism dies. So that's it for nucleic acids. I've been Mr. Johnny Hopkins, and this has been What is Biology? Thank you very much.